All right, so this is chapter 2, section 2. We're talking about water. So water has this super amazing ability to create what's called hydrogen bonding. But be very, very careful when you talk about hydrogen bonding. This is a weak, non-covalent interaction. Weak, non-covalent. So we shouldn't really call it bonds, but whatever. All right, so they have hydrogen bonds. And so it affects all kinds of different things that lead to water's unique characteristics. So we're going to look at effects on osmolarity. We're going to look at effects on cell structure, on function, and we're going to talk about the ionization of water, the autoionization of water. So we know that 70% of the mass of a cell is water. And so we are about 70% water. But what's really, really amazing about water is that it is less dense in the solid form than it is in the liquid form. So ice floats in water. This is the only substance that does that. It's crazy, but super, super important, right? And we know that liquid is water is a liquid over a very wide variety of temperatures, but the temperatures that are super important are the temperatures on planet Earth. So at most of the temperatures on planet Earth, water is a liquid, which is super important because most of our photosynthetic organisms, the ones that create oxygen that we love to breathe, live in the ocean. And so photosynthetic algae make about 50 to 80% of the oxygen that we breathe. So thank goodness water can hydrogen bond. And also it's a great solvent because of those hydrogen bonding capabilities, right? Because it's polar. And so we're going to talk about that in detail. So let's talk about that polarity of water. So hydrogen bonding occurs when a hydrogen atom is covalently bonded to an electronegative atom and in close proximity to another electronegative atom. So here's a hydrogen that is covalently bonded to an electronegative atom, and it can be nitrogen, oxygen, fluorine, sulfur. Those are kind of the big electronegative ones. So bonded to an electronegative atom. In this case, we're talking about oxygen. And so it's in close proximity to another electronegative atom. And so we see that. We have another electronegative atom. And so the hydrogen is then shared between the two. So here, this hydrogen, let me change color. Let's see if I could do that. Maybe. So this hydrogen right here, let's look at that one. So this hydrogen is basically shared between this electronegative oxygen right here, so this guy, right, and this one right here. So those two oxygens sort of share that hydrogen. It's not really a sharing because it's not really covalent. It's more of an electrostatic interaction, but you know. All right, so the bonding is very, very short-lived inside of the liquid form, right? Inside of the liquid form, the hydrogen bonds change about once every 1 to 10 picoseconds. Now, picoseconds is 1 times 10 to the minus 12 seconds. So that's like one, let's see if I count this right. 1, 2, 3. 1, 2, 3. So that's about 1 trillionth of a second, right? So they change partners very, very, very rapidly in the liquid form, not in the solid. Remember, we said the solid's a little bit different. It's unable to change as much. So this is liquid water. So when we look at solid water, the geometry of the hydrogen bonds in the water gives more space between the water and causes that ice to float on top of the water. So ice has a maximum Any given ice has a maximum of 4 bonds, excuse me, and it gives a tetrahedral shape, right? And so it gives it a lattice. It looks like any kind of lattice structure, but it's big and open. And so because of those open spaces, it's less dense than the liquid, and so it floats. And this is super, super important, because if ice didn't float, let's just pretend ice didn't float for a second. If ice didn't float, it would sink to the bottom. Well, if it sinks to the bottom, it's insulated by the water on top of it. So it's actually not going to go through the typical seasonal thaws. And then you're going to have ice forming on top of that ice and forming on top of that ice. And eventually, most of our aquatic environments would become solid ice. And, you know, then we would die because we wouldn't have any oxygen. So there are some organisms that live in places where the water is just about freezing, that it is very, very close to freezing. And they've evolved proteins that actually, instead of water being able to hydrogen bond to itself, these molecules, these proteins go in and take up those hydrogen bonds. And so the water can't form that perfect lattice, because if it does that, and you will burst the cell and the organism will die. But it's basically like their own antifreeze. And it's really, really interesting. But it just disrupts the hydrogen bonding between the water molecules. Let's see. Okay, so the other cool thing about water is water auto-ionizes, right? So you have, we say it's H+, but it's not really H+. H+ doesn't actually exist in the water. It's actually H3O+, right? And so this hydronium ion can lose a protein. Proton. It can lose that hydrogen, right? And so if that happens, and it turns into a water molecule. So here's the example, right? It starts out as a hydronium, turns into a water molecule. What's going to actually happen is that you're going to have breakage and formation of hydrogen bonds, right? It's going to break and form, break and form, break and form, until a water molecule at the end of this line, and this is showing a really short line. It's not really true, it's kind of a longer line. But that one at the end, guess what? That one becomes a hydronium ion. And so they call this the water wire property of water. So, all right, let's see if I can do this. All right. So let's talk about water's role in solubility. This is a really, really big one. So solubility is the ability of a solute to dissolve in a solvent completely, right? So think about an ionic compound. Think about, this is an example, NaCl, right? It's -1 charge and a +1 charge, so we can assume NaCl. So we have this ionic compound that forms a lattice. But what happens is the water molecules come in and they actually surround the molecules. So what you can see is the positive parts of the water are interacting with the negative ion, see that? And then the negative, the partially negative oxygens are interacting with the positive ions. And they're eventually going to surround them all, right? And so the water forms this hydration layer around the anions and the cations. And so if you think about it, this makes sense energetically. Energetically, it's favorable, right? Because when you're over here on this side, what you have is a large entropy, right? Large entropy. We've got lots of disorder going on there. But you also have a small change in enthalpy. Those strong sodium chloride bonds, ionic bonds, are replaced by lots and lots and lots of weak water interactions, hydrogen bonds, it's not hydrogen bonds, it's interactions, okay? So there are 4 types of weak non-covalent interactions. We've talked about one, we've talked about hydrogen bonding, right? So this is always going to be between hydrogen atoms on an electronegative donor and then another electronegative atom. And we talked about this. Okay, so ionic. Ionic bonds are between oppositely charged atoms. We know this. We call these electrostatic interactions, and they can actually be attraction or repulsion. And the strength of the bond depends on how far apart they are and the environment between the two atoms, right? So they work best in a hydrophobic pocket because the water basically makes a shield around the charges. And we sometimes do call them salt bridges in proteins. So like in terms of proteins, you could have an amino group, right? And maybe a carboxylic group, something like that, a positive and a negative, right? And those are just pieces on another protein. Each piece is on a protein and they can interact. And so we call those salt bridges. The other one is van der Waals interactions. Van der Waals interactions are super temporary and super weak. But they really have a big effect. So if you look at a van der Waals, that's probably about 5 kilojoules per mole, whereas a hydrogen bond is more like 20 kilojoules per mole, right? So it's much weaker than a hydrogen bond, but it's really significant in biology because you can have van der Waals interactions occurring simultaneously with great number of atoms. And so you end up with a cumulative strength that has an effect on overall biochemistry. So this is a temporary thing that occurs between dipoles of nearby electrically neutral molecules. So normally they're neutral, but they can have temporary dipoles. And so it really depends on the distance between the two atoms. There's a point at which you have repulsion because the atoms are too close. So they don't want overlap between their nuclei, right? You can't have that. So you actually have repulsion there. And then if they're too far apart, you don't have enough attraction. So there's an optimal distance between the two where you have van der Waals going on. Okay. Then you have hydrophobic effects. Hydrophobic effects occur between nonpolar molecules, and this is really cool. Remember, hydrophobic means water-fearing, right? And so these hydrophobic molecules are basically going to pack together to get away from the water, and it's not really getting away from the water. I'm going to explain what it is. But it's super important for protein folding reactions. To get those proteins in the right orientation has a lot to do with hydrophobic effects. So if you look at what happens, right? Anytime you have a hydrophobic region, so here we have 2 different amino acids and their side chains are hydrophobic. So if you look at those, there's a hydration layer, and that's this blue right here. This is the hydration layer. Here, you have it listed here. It's the hydration layer, so that's the blue. So anytime that you have a nonpolar group, what's going to happen is the water has to form this really highly structured cage. So this is like a water cage, and it has to surround the hydrophobic area. And it's really, really highly organized and structured. So you got to think about that water cage has very low entropy, right? Low disorder. It's got to be highly organized. So if those 2 nonpolar areas can come together, what you're going to end up with is a reduction in the size of the hydration layer. So if you reduce the size of that hydration layer, then what you're going to end up with is an energetically more favorable reaction. It's not a reaction, situation, right? Where you're offsetting a little bit of that decrease in entropy. Yes, you do have a hydration layer, but it's a smaller hydration layer. So that's pretty cool. You can think about it like this. So if you have salad dressing, oil and water salad dressing, right? You take it and you shake it. Just put a lot of energy into it. What's going to happen is you're going to end up with a lot of really small drops of oil inside of your water. But now you go and put that bottle of salad dressing on the counter and walk away and come back, like in 20 seconds. What you're going to see is those bubbles of those drops of oil have gotten bigger. They've come together. They've gotten bigger. And if you walk away long enough, what you'll end up with is, right, that layer of oil on top of the layer of water. And so that's the literal observation of that effect occurring. Makes you want to go have some salad now, right? Okay. So our non-covalent weak interactions play a really large role in structure and function of our biomolecules, right? So you think about enzymes. Enzymes a lot of times have multiple subunits, and those multiple subunits need to get together in order to catalyze a reaction. So if you have good non-covalent weak interactions, what can happen is those little subunits can get together easily and break apart easily. And that's kind of what you want, because you want to be able to regulate the activity of the enzymes. And so if you can just regulate that attraction based on environmental changes or exterior covalent modifications, right, then you can regulate the processes that these enzymes function in. All right. I think, yeah, okay. So then we're going to move on to osmosis. So if you think about a cell is sitting inside of an environment, right? And inside any given solution, right, you have solvent molecules, that's water, and you have solutes, so any dissolved substances inside of that solution, right? You're always going to have diffusion of the water or the solvent from areas of low solute concentration to areas of high solute concentration. And this occurs naturally, right, because of entropy. And so if you have a semi-permeable membrane, like the plasma membrane of a cell, the water can go in and out depending on the concentration of solutes. So how can the cell regulate that? Well, through osmotic pressure. So let's talk about what osmotic pressure is. So here's kind of a little tube, right? And we're going to fill it with water. And if we have equal concentrations of these solutes, so here's a solute, right? These little purple dots. And here's our semi-permeable membrane right here. That's our semi-permeable membrane. We're going to let the water through, but we're not going to let the solute through. So what happens if you increase the solvent, you're going to lead to a pressure difference, right? What's going to happen is if you started out with a low concentration here, over time, what's going to happen is those water molecules are going to move to the areas of higher concentration of that solute. And so you're going to have more water on this side. Well, if you have more water on that side, you're going to have an increase in pressure. So you're going to have a difference in osmotic pressure. So why is that important? Well, let's look at one example inside of the body. Let's get red blood cells, right? Our red blood cells change shape depending on their environment. So if you put them in a solution where you're hypotonic. Hypotonic means you have lots and lots of solute particles inside of the cell. What's going to happen is water is going to go into the cell, and it's actually going to fill the cell so much that the cell is going to rupture. Then you have the opposite, where you have a hypertonic. If you have a hypertonic, you have lots of solute particles on the outside, right? All these solute particles on the outside. So water is going to want to move out of the cell and into the solution, and they're going to shrivel and shrink, right? So over here, we had all these particles inside the cells, right? And so a hypertonic is the opposite, where we have all the solute outside of the cell. So the cell's going to shrink. But what we want is an isotonic solution. Well, how do you get an isotonic solution, right? We want to have equal amounts of water going in and going out. So what kinds of solutes are you going to have inside of the plasma of the blood to maintain that? Well, maybe you have glucose. You could have albumin, right? That's a fatty acid carrier. That's true in blood, but what about in other tissues? So all cells basically sit inside of some kind of extracellular matrix. So you have nutrients inside of the extracellular matrix, and oxygen and ions and signaling molecules. All that can help to maintain an isotonic solution to keep those cells alive and happy. So we're going to talk next about the ionization of water. So water can be ionized to the hydrogen ion, but we're not going to say the hydrogen ion, right? Well, you can, but you have to know hydrogen ion does not exist in nature, right? They're actually hydrated to form hydronium. So this is hydronium ions, right? And then if you're forming hydronium ions, you are also forming hydroxyl ions, so OH minus. So the ionization of water, when that happens, we have, if we want to know the kW of that reaction, the equilibrium of that reaction, we can multiply the concentration of H plus by the concentration of OH minus, right? So you should know this from general chemistry, 1 times 10 to the minus 14. So we can see the relationship between the two, right? So we can know, if we know this relationship, we can add any amount of acid or any amount of base, and we can calculate how much of either the hydronium ion or the hydroxyl ion is present. And we can do that, and we can get pH of the solution. So we can look at the pH scale. And so whenever we talk about pH, we're referring to the hydrogen ion concentration, right? And it's a measure, therefore, of acidity. So we know pH is a log of 1 over the hydrogen concentration, hydrogen ion concentration. So we just say negative log, right? And so pH is super important in biological systems, super, super, super important. We know pH scale is logarithmic. So if you talk about a pH change of 1, you go from 7 to 8. That's a 10-fold change in the concentration of hydrogen ions. And so a lot of things are going to go wrong when you start changing pH. And so our bodies use buffers in order to resist those changes in pH. So how do you do that? Well, you have to understand the relationship between the pH and the pKa. So this is kind of like, you have to kind of go back, right? You have to go back to your general chemistry, right? So remember, acid's a proton donor, base is a proton acceptor. So in biological systems, we use acids and we use their conjugate bases, right? So you have an equation that's like HA plus your water. I'm going to go reversible. I'm going to make H3O plus, and we're going to make A minus, right? Okay. So now remember that in this equation, we have a weak acid and a conjugate base. That's basically, so it's a weak. That's basically what your body doesn't want to use strong acids. It's not going to work. So it's a weak dissociation. So you could simplify this, right? Your weak acid going to H plus plus A minus, right? You could simplify that, even though it doesn't really occur that way in the body. But it's weak. So this doesn't really want to dissociate very much. It wants to stay together. So when you write out a Ka, right, that's your K equilibrium, concentration of H plus over A minus over, HA, right? So because the Ka for biologically relevant systems uses that same convention, right? If we're not making that much of one, we're going to use the pKa. So pKa is the negative log. pKa is the negative log of the Ka, right? Because it's not going to dissociate very much, that Ka is not going to be very big. So it's just kind of easier to work with that way. So when we do that, what we end up getting is the Henderson-Hasselbalch equation, right? That's what this is right here. And we use it to calculate the pKa of a weak acid at any given pH to see if we can experimentally determine our concentration of our weak acid and our conjugate base, and we can find out when our concentration of the two is equal. And when the concentration of the two is equal, so when these are equal, right, what's going to happen is your pH is equal to your pKa, right? And there's your buffering system. So where your pH is equal to your pKa, if you look about a pH above and a pH below, this is the buffering capacity. That's what this buffer can keep the pH at. You go farther out than that and the buffer won't work anymore. But that's plenty enough to regulate most body systems. Okay, so that is water. So we got one more section after this. I feel like I should tell y'all a joke or something while I try to shut off the recording. All right, here we go.